Okay, it is just about noon, and I do believe that we will be joined by Dr. Bradley. He was in a moment ago, and we are live right now.
I can't see him on the screen, so
Yes, so
must have had a fright run away.
Yes, we are live on Facebook at the moment, and hopefully we'll be rejoined by Dr. Bradley. But in the meantime, I'm going to go ahead and introduce myself and our guest that we have on the air right now, and I'll keep an eye out for Dr. Bradley to rejoin us. So I'm Eve Samples. I'm Executive Director of Friends of the Everglades. Every month since the advent of COVID-19, we've been hosting a live stream clean water conversation on a different topic related to Everglades restoration and water quality. And this month, we are thrilled to be joined by experts on harmful algal blooms, Dr. James Metcalf and Dr. William Bradley, who was here just a moment ago and may have had a technical difficulty. So we will admit him back into the conversation just as soon as we can. This is our fourth clean water conversation, and we do these partly to educate the public about issues pertaining to Everglades restoration, and also to keep these dire health threats in the public eye, even when they're not as visible as they have been in the past. So I know Dr. Metcalf, who I'm going to introduce, is acutely aware of the green guacamole algae blooms we saw in the St. Lucie River back in 2016, 2018, and on the Caloosahatchee side of our Lake Okeechobee. So we don't see those right now, although they do pose a threat. So we want to talk about what that threat entails, both to human health and to our environment. So with that said, I'm going to introduce you, Dr. Metcalf, and you are a senior researcher focusing on cyanotoxins at Brain Chemistry Labs in Wyoming. Beautiful place. We thank you for joining us from afar. You've been an integral part of a team that has made really groundbreaking advances into the link between cyanobacteria and neurodegenerative diseases, including ALS, Alzheimer's. And you completed your PhD at the University of Dundee, and you've been researching immunodetection and cyanobacterial toxins. And I'll just note that you have a young one next to you during this conversation, so we will be delighted if she chimes in. So welcome, Dr. Metcalf.
Yes, thank you very much from both of us. She's quiet right now, but if she has an opinion on cyanobacterial toxins, I'm sure she will let us all know.
Wonderful. Well, thank you so much for being here. Part of the reason that we were interested in speaking with you is, first of all, because of the documentary Toxic Puzzle, which many people where I sit in Stewart, Florida, and throughout the greater Everglades ecosystem watched and were concerned about. That documentary featured you and your colleagues at Brain Chemistry Labs, and I think really created new awareness about the link between cyanobacteria, toxic blue-green algae, and diseases like ALS. So you've obviously done really impressive research in that regard, you and your colleagues, but also you've been willing to speak out about the public health threats and implications and the need for better policy to protect people from these threats. And I think we don't always see that from scientists who do really important research. So your willingness to talk about it is very meaningful, very important to our audience watching today. And I just want to kick off the conversation by asking you how you got interested in this really important topic that really has become a life or death issue for many of us in Florida.
Yeah, thank you. I mean, so obviously as scientists, we're there to provide information. That's our primary goal, and our hope is to give unbiased advice. In terms of how I got involved, obviously I'm originally from the UK, so I started off doing marine biology. That's my first degree was in marine biology, and during that time I got interested in two things. One was marine toxins and venoms, and the other one was seaweeds. They're kind of weird combination, but that's what I found interesting. So I managed to get a PhD in the UK, in Scotland, in Dundee, looking at toxins produced by cyanobacteria. So cyanobacteria produce all kinds of toxins which affect the nerves, the liver. Some will act very quickly, some will act very slowly. And basically I spent 15, 16 years there in Scotland, researching various aspects from creating new methods to detect the toxins to looking, did a lot of work, for example, on dead flamingos in Kenya, because they also feed on cyanobacteria. And then around about 15 years ago, the Jackson Hole people produced a paper on a new toxin that they found in the brains of people who died of a disease called ALS-PDC in Guam. And that was a disease which contains Lou Gehrig's disease, which is the ALS part, amyotrophic lateral sclerosis, which Dr. Bradley is a world expert in. The P is Parkinson's, the D is dementia, and the C is the complex. So these people in Guam were suffering from combinations of all three diseases, and some people would get Alzheimer's and Parkinson's, some would get ALS and Parkinson's. And the thought then was that cyanobacteria found throughout the world, so this toxin might actually be a significant contributor to long-term neurological health in human beings. And gradually I got more and more involved with the groups around the world studying this toxin. And ultimately I ended up in Jackson Hole studying BMAA. This is the neurotoxin that we look at, and various aspects, and dealing with blooms all around the world.
Thank you for that. And I should relay a thank you from one of our local veterinarians in Stewart, Christina Maldonado, whom I believe you helped with a toxicology report after a dog in our area was exposed to and killed from exposure to cyanobacteria a couple of years ago. So she sent along her thank you for your assistance with that. That's
very kind of her. Thank you very much. Yeah, no, we did, and we hope to do some more work on those dog illnesses, and certainly with cyanobacterial blooms, dog deaths are a frequent occurrence, and generally act as quite a good sentinel to say, okay, something really wrong is happening in waters when you have a sick dog, and obviously it's tragic for the families involved, and yeah, it's something that we have to do. Absolutely. And I want to come back to the issue of pets and, of course, human health. But before you move on from kind of your personal story, there's a really poignant moment in the documentary Toxic Puzzle where Dr. Paul Cox, who you work with, speaks about how you all keep pictures of ALS patients up in your lab sometimes. And you really try to keep in mind the people who are suffering from these illnesses that you're working to find therapies, cures for. So he also describes a moment where you went back to the lab late one night to try to squeeze in one more experiment. So tell me a little bit about how you keep this human element at the forefront of your work. Well, I mean, ultimately, obviously, there's two aspects to the science, really. One is there is the actual interest. There's the curiosity, the fascination, the idea that there is something out there that doesn't know. I mean, cyanobacteria have no idea we're here. We are completely off their radar, but they produce compounds that are toxic to us, and that is certainly something that we find very, very interesting and fascinating. However, we're a not-for-profit here in Wyoming, and our goal is to help patients. That is, if ALS didn't exist, we probably wouldn't be doing this research. That is our reason. So if we can understand how the toxin acts upon the cells of the human body, then we can find ways to block the action of those toxicity to those cells, and those can then go forward as drugs in FDA-approved clinical trials. And we have two phase 2 FDA-approved clinical trials going on right now with Dartmouth Medical School, one for ALS and one for Alzheimer's. And yeah, I mean, Dr. Cox is a very kind and generous person to say such nice things. And I mean, yeah, we do try and do as much as possible. I mean, we have the equipment, and experiments don't do 9 to 5 either. It's not something that they say, okay, right, you can go home now because I know you've got family and things to do. But we need data. We want data. If we have to go and work late at night, so be it. I mean, it's all part of of what we do to provide information. And ultimately, as long as we can help the patients and the people, and even people who haven't developed ALS, the information that we get may prevent some people from getting ALS in the future. So time is of the essence. And with a disease like ALS, you're talking on average 2 years from diagnosis to death. So if I have to work late at night for that, that's okay.
That's very touching. Thank you. We already have a few questions rolling in from Facebook. And before we jump into those, I just want to share my screen for a moment to remind people what we're talking about. I mean, cyanobacteria and BMAA are all very technical terms, but can you see that blue-green algae there?
Yeah.
So this is the stuff that will cause PTSD for many Floridians right now, because it blanketed the St. Lucie River, the Caloosahatchee River, and a huge swath of Lake Okeechobee in 2018. I believe 80 to 90% of Lake Okeechobee's open water was covered with this bloom in 2018. So we're in better shape at the moment. We don't see this stuff on our waterways right now, but there is toxic cyanobacteria on the lake right now. I just checked this morning, the Florida Department of Health site, or DEP rather, and I think I can advance this here. This is from 2018, but these are the tests from this morning, and the red dots are just showing areas where toxins were detected. So even right now, when Lake Okeechobee is not facing a terrible bloom on the scale of 2018, there are 9 spots on Lake Okeechobee, which is a massive lake, 730 square miles, where toxins have been confirmed. And of course, that's only where they've been confirmed. So lake water is not moving east to the St. Lucie River right now, nor is it moving in great quantities west. But the risk almost always exists for us. And in our role at Friends of the Everglades, we're not medical researchers and scientific researchers like you and your colleagues are, but we do advocate for water policy that would protect us better from these blooms moving into populated areas. So that's just by way of reminder. And there's one more slide here I'm going to show. This is just this week. You can see streaks of that cyanobacteria bloom in the lake. So it's there. It's just not as bad as it was. And we're grateful for that. But we don't rest too easy. So let's talk about the near-term risks of harmful algal blooms and the long-term risks. You mentioned dogs. We were talking about dogs earlier, and I believe that pertains to more of the acute risk of ingesting this stuff. But can you kind of break it down in terms of what the potential health threats are for people and animals?
Sure. I mean, animals is a bit more, what's the word? Animals jump into water. That's really the big problem with things like dogs, is that they seem to like the smell of cyanobacteria. As we've probably all seen in the past, dogs tend to end up in areas with smells that we don't like in the house. And it's the same as cyanobacteria. Cyanobacterial blooms really can smell quite badly. So that's really the risk with dogs, is that they're attracted to that smell. They run in, they lap at the water. If there's, say, mats on the bottom of the lake, they will eat those mats. The cyanobacteria can get stuck to the fur. So when the dogs come out, they clean themselves and they can get further intoxication. So really dogs, well, we started to understand perhaps more in the fact that dogs are certainly at risk of short-term effects and perhaps long-term as well. We're starting to do some research into that. But in terms of cyanobacteria, there's a whole host of things that can be of a problem. As you mentioned, we tend to split the toxins into classes and also into terms of action. So there are toxins that act short-term and there's toxins that act long-term. In terms of the short-term ones, there are the neurotoxins that they produce, like anatoxin A, saxitoxins, anatoxin AS. These are very, very, very powerful toxins. They have actually been used as suicide capsules historically because they are extremely potent. And you're talking probably, I don't know, 20 minutes from ingestion to a point of no return.
Wow.
Then there's the microcystins, which is what we've been seeing in Florida a lot of. These are small proteins, basically, and in high enough doses, and certainly what we've been studying in Florida on the St. Lucie Canal, and we actually also have our data on the Caloosahatchee from 2018 will be coming out in the next few weeks as well. We're seeing very, very high concentrations of microcystins, these peptide toxins, which is really what the major risk is short term. And in a high enough dose, you're probably talking an hour or two for death to occur. And those work really by, and it sounds horrible, is that they basically dissolve your liver and you bleed into your liver so that the rest of your body has no blood to function. That's really how they work.
Wow.
So those are the real, short-term risks that we have dealing with. Long-term risks, these are evidence that suggests that microcystins themselves can cause they can enhance the risk of primary liver cancer. They're what we call tumor promoters. So if you had, say, some compound in your diet which could create a tumor, then microcystins will speed up that process of creating cancer in the long term. And also we have another long-term toxin, which is BMAA, which is the ones that we've been dealing a lot with, which we've found in the brains of people that died from Alzheimer's, ALS, Parkinson's, and we're linking those neurological diseases with exposure to cyanobacteria, and Dr. Bradley is kind of the expert on that. But also cyanobacteria these blooms are ecosystems in their own right. We have found, other bacteria in the bloom that live on the bloom that could be of significance to human health. And certainly, as everyone that lives in Florida and near these blooms is aware, people report a lot of symptoms that they don't like. I got a headache, I got sore throat, I can't breathe very well. So there's a whole host of things that may be happening in terms of the bloom. And certainly with things like BMAA, we think your genes, your genetics have some effect. So let me backtrack. With the short-term high-dose toxins, your genes won't really make any difference. If you have a high enough dose of microcystin, you are going to get very sick and perhaps die. But when you have the long-term toxins, we're talking about exposure to much lower concentrations of the toxin over a much longer period of time, and then perhaps your genetics become important. Maybe you're completely immune to the action of BMAA, or maybe you have a gene which predisposes you to Alzheimer's or to ALS, and then you're the ones that are at risk. And certainly we're trying to understand what genes are involved with susceptibility to the action of these toxins. So it's certainly a risk that we want to understand, and it may be that some people don't have a risk, whereas other people are likely to get ALS or to get Alzheimer's. We haven't seen any of the real problems that we've seen in other parts of the world. For example, like in Bangladesh, when you have blooms of cyanobacteria, you can get outbreaks of cholera because the cholera bacteria can live on the cyanobacteria. So there's a whole host of things that are going on. Generally with the scums, we tend to, say, avoid the scum. Don't go swimming in the water with scum because it's probably not going to be good for you. And then obviously with the dogs, so with people, you can say to a person, avoid the scum, don't go near the scum. But with a dog, you can't say, Fido, don't go near the scum, because the dog's going to go running into the water, lapping, and then get intoxicated by that.
Right. Thank you for filling us in on that. And I believe Dr. Bradley has rebooted and is joining us now. Dr. Bradley, can you hear us okay? There you are. We see you too. Welcome.
I apologize for the communication problems.
No problem at all. We are live on Facebook and talking about harmful algal blooms and the health risks. And we appreciate you being here. These technology glitches are sort of the nature of the era we're living in right now. So no worries at all. It's a good timing for you to rejoin us because Dr. Metcalf was just explaining the range of health risks posed by harmful algal blooms, ranging from very acute liver disease to long-term implications related to ALS, which of course you are a leading expert on. So I wanted to give you a moment to talk about work you've done on ALS in relation to cyanobacteria and harmful algal blooms we've seen in Florida. And you live in Florida, you're in Miami, you're not far from where we've seen really alarming blooms on the ground. So please fill us in a little bit on your work and what hope we can have that we're nearing a cure.
Yeah, so my work in fact first started when I met Paul Cox, and with him studied what he was doing with regard to cyanobacteria and the Guam ALS focus. And then I followed through with him and with his colleagues, including James Metcalfe, of what they've been doing about proving that BMAA is a cyanotoxin, is a toxin produced by cyanobacteria that can produce things that look like ALS in animals. So, and then I joined with Elijah Stommel, who's a neurologist in Dartmouth, New Hampshire, who had identified clusters of ALS patients that were adjacent to lakes that had frequent algal blooms and had really quite a high incidence of ALS. And since that time, we've been advancing our work in terms of trying to tie down the frequency of ALS adjacent to areas where there are frequent algal blooms, both in the New England area, which is where Elijah Stommel is, in Ohio and in Florida. That work is still in progress, so we don't have anything definitive to really be able to say that we've proven that ALS can result from exposure to, that is chronic exposure to these cyanotoxins, but we're moving along in that direction.
And if I could interject there, I'll just orient the audience to that you're a professor and chairman emeritus of the Department of Neurology at University of Miami. I want to give you that portion of a proper introduction. But I was really struck by an op-ed that you co-authored earlier this year about what you just spoke on. So we have many indications about the link between these toxins that are in blue-green algae, which we've seen in our waters, and ALS, Lou Gehrig's disease. We don't have ironclad, indisputable proof, but you wrote an op-ed arguing that we shouldn't wait another 20 years to find out because the indications are strong enough that we should take public health actions now to be preventative. Talk a little bit about the role of our public health leaders and why you've ventured into that debate.
Yes, so what I drew in that op-ed piece was an analogy with smoking, and it took us 30 years, in fact, to really determine that smoking was a cause of lung cancer and to force the tobacco manufacturers to put warning labels on the cigarette packs. And we are about in that situation now with regard to ALS and the cyanobacterial blooms. We know that the cyanobacteria produce a lot of toxins, some, as James has said, that are acute toxins, and some that have certainly been pretty well linked to the long-term frequency, long-term risk of getting liver cancer. We've got really good evidence that the cyanotoxins can produce in animals the changes that are like Alzheimer's disease and ALS. And that's too good a linkage, from my point of view, to say to the public health people, you must take notice of this and you must warn the public of it. And that was the point of the ALS. What we were asking for was to have public notification when there are algal blooms, not just to warn people about the acute effects, but to warn about the risks of potential long-term effects.
Right. The lack of communication and notification has been a major problem in Florida. I do want to ask Dr. Metcalfe about that too. During the 2018 algal bloom in Southwest Florida, just getting basic information from the Florida Department of Health was a challenge for residents. And I do think there have been some incremental gains on the part of the State Department of Health and DEP, but the Waterkeepers in Florida, Waterkeepers Florida Alliance, did a survey recently of signage and found that even in areas where there is toxicity confirmed, there's not always signage today. So there's really more progress, more work that needs to be done in terms of plans for preventing the public from coming into direct contact with these algal blooms in Florida and beyond. I mean, the problem exists in Lake Erie. My family goes to Lake Erie in the summers, and I've seen it in the waters there, and there are no signs in many places. So to that end, Dr. Metcalf, can you talk about the paper you published with your colleagues in 2018 that made some pretty explicit recommendations in terms of what our state government leaders and federal government leaders, I believe you addressed to, can do to help the public, protect the public?
Yeah, I mean, obviously, as we're seeing with the COVID outbreak, science is a very, very important aspect to life, and science is there to give information and provide recommendations and other things that don't have to be, they can be ignored, but it's kind of, you know, this is what we understand, this is what we know, this is where we think we should be going. So certainly signs are a big issue, and you know, talking to various groups like people like Bull Sugar, you know, they're the ones that they actually paid for warning signs on the St. Lucie River, the St. Lucie Canal. So that's important, but people also get blind to signs. If you have a sign, if you have a bloom from beginning of July to the end of August, and there's a sign saying beware blue-green algae or cyanobacteria, people do become complacent. So there has to be some kind of way of saying, look, don't go into the water at this point in time because it's very, very high. We have spoken to the department, I think it was Environmental Health, I can't remember exactly which one it was. And they actually agreed and said, well, we assume the scum is toxic. And they measure the water, the concentration of algae and cyanobacteria in water in places where it's not quite as concentrated. So ultimately, we need to have legislation to say the water has to be of a certain quality. Whether it's specifics, say, you know, microcystins cannot be greater than 1 microgram per liter, which is a drinking water guideline from the WHO, or whether you have a blanket sort of guidance which says you cannot supply water injurious to health, which is certainly what happens in other parts of the world, is that you can put cyanobacteria into a category whereby if you know it could hurt you, then you should do something about it. There's also different issues concerning drinking versus bathing versus recreational waters versus fishing waters. I mean, it goes on and on. I mean, we saw a lot of people fishing in blooms of cyanobacteria in 2016, and people are eating those fish. And, you know, it's something that I would certainly think twice about. I mean, when you cook your fish, you don't destroy the toxins. The toxins are extremely stable and will survive in the fish tissue. So at the moment, the best, I think, and as you mentioned, it is a worldwide problem. So Florida can sort this problem out. It can be solved and it can be got rid of. Ohio probably is the best state in the US currently in terms of dealing with cyanobacterial toxins, largely because of, you know, Lake Erie.
Let me just pick up on what James said, because indeed in Lake Erie, the water supply, the domestic water supply for Toledo and another city adjacent to that was shut down in 2016, 2017 because of high microcystin levels in the water. And they do have major algal blooms that occur in that area. In fact, that's been a focus of our research, looking at cyanobacterial content of the water and looking at ALS frequency in the state. There certainly are major areas of risk, not just in Lake Erie, but in the other lakes that are around there. So it's for your family, Eve. Yes, I would have concern, and really, it's exactly the same situation here in Florida. We were able to show in 2009, 2010 that there is a food chain accumulation of BMAA, the cyanotoxin that Paul Cox identified in Guam. The BMAA accumulates from the cyanobacteria into the crabs and shellfish and into the fish. And then we have shown that there is BMAA in the brains of ALS patients, Alzheimer's disease patients, but not in control patients. And so there's that linkage again between the research on the cyanobacteria and all the way through to human disease. And that's another reason that really the public health officials should be taking notice of all of the science that James and Paul Cox have been putting together.
That's really valuable. And I think the distinction about the food supply, the food chain is really important because it shows that this impacts all socioeconomic classes. If you're a subsistence fisherman, you can be harmed by this. If you live in $1 million home with a yacht in your canal, you can be harmed by this. And there's some indication that these toxins can be aerosolized. And if you live a mile or two away, potentially you could be harmed. So these are serious concerns and they should be taken as seriously as possible by our government agencies. And I'll just add that Friends of the Everglades has supported efforts to set a limit on microcystin that's allowed to be discharged from Lake Okeechobee into the St. Lucie River. This is something advanced by our local Congressman, Brian Mast, and the limit there would be 8 parts per billion, which is in line with the recreational limit. So I do think those hard limits can be useful. In the interest of time, though, we've been talking mostly about the effects of these toxic algae blooms, but let's talk about the causes. We're getting some questions from Facebook about the causes. And one of the questioners asks if overuse of the herbicide Roundup, which is used frequently by Fish and Wildlife Commission, could be contributing to the problems of harmful algal blooms. So nutrient pollution obviously is a problem too. Can you talk a little bit about what's causing these blooms and why we're seeing them more often? I'll open that up to either one of you.
James, do you want to start?
Sure. I mean, yeah, you're right. Nutrients are the basis for cyanobacterial blooms. Cyanobacteria just need a few things. They need nitrogen, they need phosphorus, they need some carbon, and they need sunlight. I mean, that's really what they need. In terms of what's going on in Florida, there's a number of, how do I say this? There's a lot of finger pointing going on about who is causing the pollution. And I still think that there's multiple sources of pollution occurring that's creating these blooms. You know, there's talk of agriculture, septic tanks, urbanization, and I think all these things have parts to play. And certainly, nutrient control is certainly something that needs to be done. Florida itself its bedrock is phosphorus. So I mean, there's a problem there in the fact that you can't remove phosphorus because Florida is phosphorus. So it's going to be nitrogen control that really is the issue that deals with those blooms and gets rid of them. You can do things like green livers. You can have, I mean, the Everglades themselves would be a perfect solution to the problem. If you had the Everglades, there's lots of plants, trees. There's a large, wide spread of water flowing from north to south. So really, that is the ultimate solution, is to recreate the Everglades and get rid of the nutrients. That's really the issue. And certainly, you know, back pumping from Lake Okeechobee up to agriculture and back again, you're picking up nutrients all the way. So it's a long-term, expensive, but solvable problem. You need to get rid of those nutrients, mostly nitrogen. And if you do that, then the bloom should go away, or certainly subside to a point whereby they don't cause too many problems. And we have to remember cyanobacteria are, you know, they're 3.6 billion years old. They're everywhere. They are part of the ecosystem. So we are not going to remove cyanobacteria from Florida because that probably would be a bad thing anyway, ultimately. But we can certainly get them to a point where they're manageable.
And to the point about Roundup and herbicides, is there any role there?
So, I mean, yeah, I've been asked that question many times in terms of, you know, is glyphosate a problem? I've seen reports where it's good for blooms, bad for blooms. My personal feeling, and this is, I need to do a bit more research, is that if Roundup affects higher plants more than it affects bacteria, because cyanobacteria are bacteria, they're not actually plants like you would find in your garden. So it comes down to which are more susceptible to the herbicide. If cyanobacteria are more resistant because they're a bacterium rather than being a plant, then they'll do just fine in the presence of Roundup and actually will probably get more nutrients available for them to grow. If they're more susceptible to Roundup, then it's certainly likely to affect the bloom. But I mean, we know from court cases that glyphosate is a carcinogen, and there's all kinds of things that are going on. And ultimately, natural solutions like the Everglades are a far better remedy than adding chemical A to environment B and seeing what happens, and then trying to deal with the downstream issues that occur then. So ultimately, it requires stepping back, getting the information, having a political will to actually say, okay, do we want to deal with this problem? And if so, how are we going to go about it? And really just no sort of half measures and saying, okay, let's get rid of the nitrogen. Let's find out where the pollution sources are, deal with those pollution sources, and then, it'll take a bit of a while, but hopefully then things will start to become more balanced in the environment.
Mm-hmm.
Okay, let me add a couple of things, if I may, to what James has said. So going back to the question of what are the sources of the nitrogen, a lot of the developments, and obviously Florida is a state where there's an increasing amount of development, too many people for a limited area of land that is available. The majority of those developments work on septic tanks, and many of those septic tanks are actually old and are leaking. And so that's certainly a source of pollution that gets into the environment and gets into the water bodies. More of the water treatment plants also need to be expanded. And that's really what James was hinting at, that in order to get rid of these things, there needs to be work on the septic systems and work on the water treatment plants that are the basis of human development in those areas. And then turning to the source and the relationship to the Everglades, these massive blooms that occurred in 2016-17 were the result of a major releases of water from Lake Okeechobee that came because the Corps of Engineers was fearing that a hurricane would bring in such a lot of water that the dam would collapse. What would do much better, in fact, is if there was a graded release of water, which they keep up because of agricultural demands and agricultural lobbying of the legislature. They keep up so that there is irrigation of the agriculture. And if there was a graded slow release over the whole of the summer period of rain in this state, down into the Everglades, that would certainly, as James was saying, result in a very much reduced amount of nitrogen flowing out into the water on both sides of the peninsula. And then finally, with regard to Roundup, to my mind, the linkage is not in any sense known, but I will add that toxins can add one onto the other in producing damage to cells and damage to animals and humans. And therefore, if Roundup is a toxin, then the addition of that to the cyanotoxins is certainly not a good thing to have.
Great point. To your point about how water from Lake Okeechobee is managed, we're seeing some incremental progress and open-mindedness from the Army Corps of Engineers on that front. We just are drafting comments now about, pertaining to the Corps' deviation of the Lake Okeechobee schedule. So it would call for slower releases when there are no cyanobacteria blooms or any harmful algal blooms, and ideally no releases when they do exist. So the Corps is ever so slowly making progress, but we have a question about the Corps here. This one comes from Jackie Lopez from our friends at the Center for Biological Diversity. And she's asking if you could respond to the Army Corps of Engineers statement that, quote, "there is no known linkage between BMAA and neurodegenerative diseases." They cite Menely et al. And it says BMAA is not found in the brains of patients with confirmed Alzheimer's disease. So we spoke about this debate a little bit, the lack of ironclad confirmation. Can you respond to the Army Corps asserting that?
Yeah, can I do that, James? Because
Please, Walter, yeah, go ahead.
I've played a sort of a peripheral role in watching all of this. Most of it comes out of the laboratory that James works in, the Paul Cox's laboratory, and his colleague Sandra Bannock. The paper that is quoted by the Army Corps of Engineers is in fact one of a small number of studies that could not find BMAA in a variety of different things that they looked at. And they were not using the same assay as was used by Paul Cox and Sandra Bannock, which really is now the gold standard for the assay of BMAA. The result of that is that they are cherry picking research that says negative, while in fact there is a major body of work by the more accepted scientists that says there is a linkage between BMAA and the brain and BMAA produced by cyanobacteria. So I think that this is an example of really biased cherry picking of research, and I really condemn them for it.
That's very clear answer. Thank you for that. Would you add anything, Dr. Metcalf?
I mean, yeah, it's scientific research is about being given a question. I mean, we are pretty ignorant, and we are trying to find answers to questions that we're asking. So we're trying to ask the question, is there a risk from the environment of developing diseases like ALS and Alzheimer's disease? We know, and as Dr. Bradley said, that the techniques that we are using are the ones that the analytical chemists organizations think are the best methods to use. So we know that BMAA is present in the environment. There have been many papers published from many groups around the world saying that if you go to an environment where cyanobacteria are, you can find BMAA. We can find BMAA in the brains of people who died of ALS and Alzheimer's disease. So there is definitely a connection there. And the work that our colleagues at the University of Miami are doing have shown that we can create the tangles and plaques in the brain and in the spinal cords of animals when you administer BMAA, and we know ways of stopping it. We have clinical trial data which says that administering L-serine seems to slow the development of ALS. So one study can certainly be an issue in terms of, preventing research happening, but the actual body of research is like it's a massive boulder that's slowly rolling down the hill. And it's picking up lots and lots of information as it rolls down that hill. And at present, I would say the body of evidence is that BMAA is a risk factor for ALS. And as Dr. Bradley said, with smoking, you can smoke for 45, 50 years and not get lung cancer. You can work in a bar where someone was smoking next to you for 20 years and end up with lung cancer, no problem at all. So we know that at these low chronic doses, your genetic makeup becomes very, very important as well. Mm-hmm. And in terms of diseases like ALS and Alzheimer's, we've only found one natural toxin that can perhaps cause these diseases. There are probably others as well. We know that you can make there's a compound called MPTP, which can cause Parkinson's in people. So we don't like making blanket statements to say, it's completely you cannot get ALS from being exposed to BMAA, but our research says that it's something that you should pay attention to because you may end up with ALS or Alzheimer's disease. Mm-hmm. Is that fair, Walter?
Yes, I think so. And just to say, the CDC, which is receiving a lot of both support and hammering from the current administration at this present time, the CDC has been very good to be interested in the research that we've been doing with regard to the association of cyanobacteria and ALS and Alzheimer's disease. and have in fact given us 3 grants over the last 8 years that have advanced our research in this area. So the CDC certainly recognizes the need for research in this arena.
I want to go back to the metaphor Dr. Mcafee used of the giant ball rolling downhill and the body of research. And I think we can say safely that there are some powerful interests out there that don't want environmental toxins linked to these scary diseases, because there are some powerful lobbies that contribute to those toxins and the creation of those toxins. Have either of you ever felt pressured not to pursue these links? And if so, how did you react?
I personally,
go on,
Walter.
Well, from my point of view, just the difficulty of getting research funds to do these, the research that is necessary. There's never enough money for research, but it is not, it's been an uphill battle, to turn James's analogy around, to get funding for the research. But so far we've been successful. And in terms of pressure, and what I will say is that there has been back pressure from the authorities in the health arena in Florida at any rate, to resist putting up notices about the potential long-term risks. And that's certainly been a kickback that we have seen. Mm-hmm. And I think the latest is this thing that you mentioned from the Army Corps of Engineers. So to that extent, that's a kickback.
Yeah, I mean, I agree with Walter that scientific research does require funding, and it has been difficult to get funding. I personally have not had any pressure to not pursue this research. It's definitely, and certainly the way that the brain chemistry labs works is that we're free to pursue whatever we think is worth pursuing. My interactions with like the Army Corps of Engineers have always been very good. We were out at Lake Okeechobee, and there's an Army Corps of Engineers out on the boat, and they came over and they said, "Do you want a sample?" And we said, "Thank you very much." And we had a nice chat with them for 15, 20 minutes. So people generally want to solve the problem. And obviously, I mean, I think it's going to but organizations like ourselves, we're low down compared to political pressure. That's going to be higher up the chain, I think, where that might occur. And another thing is we live in Wyoming, so it gives us a nice distance away from Florida, so that we don't have the same kind of pressures that perhaps someone in Florida might get, because we're collecting our samples, we're analyzing the data, send the data off, working with people like the water keepers. And what we're seeing, I think, also in Florida is that the people now are starting to resist and say, you know what, I'm not interested in what political affiliation you have. I just want to have clean water. Mm-hmm. And I think that message is starting to kind of get through. And the data is the data. I mean, we're not making things up. We have no interest in creating data for data's sake. The data suggests that exposure to cyanobacteria might give you a disease that you do not want to have later in life. Mm-hmm.
Yeah. So, Eve, you might talk about the governor's task force on clean water in the state, and the what was eventually the outcome in terms of the bill that the governor did sign.
Sure. So to your point about outrage on the part of the public and political will, we did see some positive progress in that regard in 2018, because conditions were so bad. I think in no small part because of that outrage, DeSantis was elected and quickly rolled out an executive order on the environment. One of the provisions of that executive order was creation of a blue-green algae task force. And that task force hasn't met since last fall now. And of course, we have very pressing public health matters to tend to. But what's a little bit more discouraging about that is that the recommendations of that task force, many of them were not incorporated into the so-called Clean Waterways Act that was passed by the Florida legislature this past session. Florida legislature has really been part of this problem. I don't think we can say it any other way. There have not been enough bold advocates from either party in Tallahassee, and that really needs to change. So we're hopeful that DeSantis can create some more positive action to embrace that task force's recommendations going forward. And they're pretty basic things. And then we really need to get at the sources of the nutrients that are leading to these blooms, to your earlier point. So thank you for the opportunity to weigh in on that. We've got some more good questions here. And this one I've wondered about too. The question is, what happens when we irrigate vegetables and sugar crops with water containing cyanobacteria?
Well, it's, let me backtrack. Many years ago, we actually had in the UK, we had a farmer who was growing lettuces, and he had a brick pond, and that brick pond ended up with a bloom of Microcystis, which is the same organism as we have in Lake Okeechobee, producing the same toxin, Microcystin. And he spray irrigated his lettuces with the bloom of Cyanobacteria. So we analyzed lettuces, and they were too toxic to eat. And the farmer did the right thing and said, okay, fair enough, I'll just destroy my crop. So there is definitely a risk. We know that microcystins can contaminate from spray irrigation. It's hard to say what you mean by in the water. If it's in as a free compound, then there's a possible taking up in the roots. We know that BMA can go into the roots of things like wheat. So yes, there is definitely a risk of spray irrigating plants and vegetables and crops with cyanobacteria, because the toxins probably will be on those plants, and without testing, you don't know what you're consuming. It's the same as pesticides and things like that. So there is definitely a risk, and we've known about that for about 20 years.
Yeah, just to add to that, there are papers in the literature that demonstrate the accumulation of BMAA in plants that are growing in water that's contaminated by cyanobacteria.
That's helpful. Another question, do scientists know how to remove BMAA from our drinking water yet? I know this came up in the Toledo area.
The evidence that we have so far suggests that if you have a good water treatment plant, you probably will not have BMAA in the water. Sand filters seem to be, according to the literature, the best way. And sand filters work by, you kind of want to let them mature, so they have lots of bacteria inside the sand filter, and that seems to be what takes up the BMAA. So my personal feeling is that a good water treatment plant will protect you from most toxins.
Okay, now I'll jump to the next question. This one relates to the threshold for risk. And I know genetics play a role, you both touched on that, but so many people want to know how much exposure is too much exposure. So this person is asking about the risk to commercial fishermen who are on the water 250 days a year, 10 to 15 hour days. What can you say about the risk posed to a group like that? Yeah,
so this is really difficult because let me draw the analogy between head injuries and disease. People have known for many years that people who are professional boxers have a risk of developing dementia gradually. But that is develops 25, 30 years after they give up their professional career. This relationship between time and the development of the disease is really a confounding and a difficult thing in regard to the research. So the same goes in terms of cyanobacterial toxin exposure. So, and then finally you mentioned, and your questioner mentioned this question of genetics. There is no question that obviously as James alluded to earlier, somebody can smoke for 50 years and not get lung cancer. Somebody else can smoke for 10 years and go down with lung cancer. So they obviously have a genetic predisposition to getting these. That's in fact the topic of our latest research grant, is looking at the genetics of the individuals, ALS patients and control patients, looking at all of their genes, and then looking at what's the risk for control patients and patients who get ALS in regard of the mix of genes that they have and the exposure that they have to various environmental things like cyanobacteria. So this is a field of very active research, but it's also a field of very complex research.
We got another really good question ahead of this conversation about the risk of dredging areas that were impacted previously by cyanobacteria blooms. So the St. Lucie River, which you're both familiar with, which had a terrible algae bloom in 2018 and 2016, will be dredged by the Army Corps for navigation in the next year or so. Is there any risk of stirring up microcystin or renewing the risk to microcystin, even if it's not visible via a dredging project?
James?
Yeah, I mean, it's a very interesting question. We have to sort of step back a little bit. Historically, when we've had a lake with a history of cyanobacterial blooms, we can drain that lake, remove the sediment, and you will not get recurring blooms because you're removing nutrients that are present in the sediment. And also, as your question alluded to, you can have parts of colonies that overwinter in the sediment. So in that respect, the St. Lucie River Canal, if you dredge that, you would remove potentially any nutrients and cyanobacterial cells that are there. However, it's a river, not a lake. So the cyanobacteria don't like growing in rivers because they don't like fast water. They like sort of still water where they can bloom gently and cause big problems. So my feeling is that dredging the St. Lucie Canal will probably not do harm to the future. However, and I was talking to my partner last night about this, what is the cost of dredging the St. Lucie Canal? And in that respect, could that money be put to better uses to deal with the problems of the blooms? I appreciate if navigation is an issue, then you have to dredge, but they'll certainly stir up things. And if they're dredging, are they going to put the material out into the ocean? Are they going to put it onto barges, take it to landfill? So in the past with blooms and bloom sediments, they have been classified as toxic waste in other parts of the world. So it's sort of like the risk question. There are so many unknowns that we don't know. My feeling is that by dredging the lake, you're probably going to cause less problems in the future, but the blooms are not starting in the river, they're starting in Lake Okeechobee. So if the blooms in Lake Okeechobee, you're going to release that bloom down the St. Lucie Canal and it's still going to be there. Maybe less nutrients, but I don't think it'll be worse in the long term. Short term, you might get some release of heavy metals, and certainly we see that, for example, in dams throughout the world. If you have a dam in a river, you end up with large concentrations of heavy metals at the dam wall because they don't go through. You get evaporation and all those kind of things, and we have lock gates. So has anyone actually looked to see what the sediment contains when they're dredging and how far they're dredging? Are they dredging all the way up to Port Mayaca? I mean, it's a can of worms, effectively. Yeah. But there's, as the basic answer to your question, probably not going to cause it any major problems to start in long term.
Very good. I have a couple more topics I want to hit on in our last few minutes. One is this pandemic that we're all living through right now, and two is I'd like to end on a hopeful note about your research toward a cure for ALS or therapies. So let's start with the pandemic. We've all been living through extraordinary times. I'm so encouraged by people who follow Friends of the Everglades and still have the mental bandwidth to think about these environmental issues we're discussing. And I think that's partly because they're not unrelated. This is a public health threat, everything we've just discussed, and COVID-19 is a public health threat. So Dr. Bradley, you and I touched on this when we spoke last week, but how do you see the relationship between these two health threats?
Well, they seem in many ways to be separate. They're both of them have to be run through public health agencies, and we've alluded to that already. But nevertheless, if you're in a sick state, if your health is not good, then we know that, in fact, you're at much greater risk of the bad effects of the COVID virus. The elderly, the people with various other diseases, the disadvantaged. These are the people that, in fact, get the disease more severely. And to that extent, if you happen to have severe exposure to cyanobacterial toxins, then in fact it is quite likely that you'll have more risk of getting the severe effects of COVID-19.
Okay, that's very helpful context. Thank you. So not a direct relationship, but certainly it could amplify risk just because of a compromised health condition. Okay, and before we get to the therapy, somebody has asked a couple of questions, and this is very narrowly focused, but it's for Dr. Metcalf. Is there any evidence that the elephant die-off in Botswana is a result of cyanobacteria toxins?
Not that I'm aware of. However, we have certainly in the past seen, if I remember rightly, hippo deaths in African national parks. We've had flamingo deaths in African national parks. So certainly it is not infeasible, but I am not aware of any research suggesting that the elephants have died from that. And certainly with water, there's all kinds of waterborne diseases that could floor an elephant.
Thank you for addressing that. So let's end on the progress you've made. There was some encouraging news earlier this year about L-serine potential therapy for ALS. Can you both weigh in on how far off we are from a cure for some of these neurodegenerative diseases you've researched?
So let me start with that. The work that Paul Cox did in terms of the animal research of exposing vervets to BMAA was able to show that this amino acid L-serine is able partially to block the toxicity of that. And that came from work which was done in cells in tissue culture by Rachel Dunlop, who was in the lab with James and one of her colleagues, Ken Rogers, in Australia. In these vervets, it is possible, as I said, to block the effect with L-serine. And from that came the first, what is called a phase 1 trial of L-serine in patients with ALS. We were able to show in a small number of patients that there appeared to be a dose response effect. That is, the higher dose of L-serine seemed to produce a greater slowing down of the rate of ALS. Now, that has now been taken into 2 phase 2 trials, one in ALS and one in Alzheimer's disease that is being undertaken in Dartmouth Medical School in Dartmouth, New Hampshire. Those trials are still in operation, and we've probably got another year before we will know the outcome of either of those trials. So these are all very encouraging, but obviously there is a very long history of failed trials in ALS and Alzheimer's disease where we've had hopes and in the end they've not produced the outcome. So we don't want people to get overly optimistic about this. We as scientists have to maintain our reserve with regard to whether this is going to work or not going to work.
Thank you. Anything to add to that, Dr. Metcalf?
No, I think Dr. Bradley sort of, said it all, is that our research is very promising. We will continue as long as that research is promising. If we can prevent people from suffering from ALS or give them extra years, then we'll continue to do that as much as we can. If we find that L-serine doesn't work, then we will go and find something else that does work. So I suppose our hope is that we will find a cure because we will keep on trying.
Thank you. And in the meantime, preventing widespread exposure to these toxins is really the best route. And even maybe after a cure is identified, that will still be necessary, I suspect. So that means better water policy, better public health policy, and better attention to the important research that you all are doing. We very much appreciate your time today, sharing your expertise on this important topic. We hope you'll keep in touch, and thank you again.
Eve, thank you.
Thank you very much, Eve.
All right, take care.
Be well.
You too.
Bye-bye.
Bye-bye.
Bye.